Everybody. Thank you for all joining us. My name is Vrije, and I work at the Department of Psychiatry and the Center for Brain Health at the University of British Columbia. I will be your moderator for this event today. Welcome to the seventh event of our educational program. Before introducing our guest speaker, Ilan, today, I will have a quick introduction of the educational program. So with this program, we aim to progress the translational application of computational neuroimaging techniques to enhance human brain health. This educational program is nested within lifespan working group of the Enigma Consortium. It is a very inclusive program that we welcome researchers at all levels of neuroimaging skills and from diverse training backgrounds. The directors of this program are Dr. Shalala Haas from Mount Sinai, New York City, and myself, and we do have an international advisory committee. Please consider to join our mailing list to receive a notification about the updates of this program. We have a YouTube channel, and there you can find videos, video recordings from our past events. We also have a Twitter, now called X. Please consider to follow it there. And if you wish to share your work through this platform, and please get in touch. We would be very happy to host you as a guest speaker. Before I pass the microphone to our today's speaker, I would like to mention that we have two events scheduled. The first one will be on December 1st, and we will have Yutong, Yu, and myself to introduce our team's recent work on central brain normative modeling and brain age models. Please know that this one will be presented in Mandarin, and we already have an English version. Well, you can find it in our YouTube channel. So if you happen to speak Mandarin, please consider to register for it. We will also have Dr. Lindsay Oliver and Salim Mansour from Toronto. They will be presenting their work on using neuroimaging techniques to localize neurostimulation targets in the brain. The date and time of this event are yet to be finalized, so please stay tuned for updates if you're interested in this topic. Today, I'm very delighted to introduce Ilan Libedinsky and thank him for agreeing to give us a talk. Ilan is a PhD candidate for Virginia University, Amsterdam. He will be presenting his recent work on polyconnectomic scoring, which is a novel method developed to quantify whole brain functional connectivity patterns associated with brain disorders. This work was recently published in Biological Psychiatry, and we believe it marks a significant contribution to the field. So please join me in congratulating Ilan on this big achievement and welcoming him to share his insight with us. Ilan, now the microphone is yours.
Thank you very much for your kind words. I will now share my screen. Okay, everyone can see the screen, right?
Yes.
So hello, everyone. As Ruyang said, my name is Ilan Libedinsky. I'm a PhD candidate at the Vrije University, Amsterdam, in the complex traits genetic department under the supervision of Martijn van den Heuvel. And I'm very excited to be here today to share a tool that we've been developing over the past year called Polyconnectomic scores. So today's goal is to explain the rationale behind this tool, the method involved, and its various applications, so everyone can use it in their own studies. Okay, so let's start a bit with the motivation behind this study. Historically, both genetics and neuroimaging fields began their effort to understand the neurobiology of brain disorder by focusing on a specific candidate genes or brain regions, selected a priori based on a specific hypothesis. Now, over the past decade, larger samples has been acquired, enabling more comprehensive investigation of the brain. This shift has allowed us to start studying specific white matter tracts interconnecting brain region and move even beyond toward a more extensive analysis of whole brain connectivity and network organization. This has enabled the detection of network properties altered in disorders, for example, and also the detection of subtle effects that were previously unreported. However, very weak effects may still go undetected in a typical neuroimaging study. It is still possible that there are widespread, diffuse, and subtle alterations in brain disorders, yet these alterations are not being effectively identified or utilized for further analysis. So we aim to develop a method that allows us to go beyond investigating just the tip of the iceberg, in this case, individual connections or edges, or group of connections that would be a cluster network, and move towards a deeper analysis of the global whole brain alteration that potentially are happening in brain disorders. So to achieve this, we took inspiration from polygenic risk-scores in the genetic field. In genome-wide association studies, GWAS, individuals from two groups, such as patients and control, are compared to identify genetic variants associated with a phenotype of interest. Now, all risk variants associated with the phenotype that are present in an individual's genome are then aggregated into a single score summarizing the genetic predisposition to that phenotype. Okay, so in our case, we want to from this method and apply it to the field of connectomics. So our goal is to generate one score per subject that indicates the extent to which brain signatures associated with a disorder are present in an individual. Okay, so to achieve this, it is first essential to identify the characteristic brain alterations associated with a disorder. These brain alterations serve as a blueprint, let's say. We then overlay an individual's brain data into this template, calculating the similarity between the individual's brain pattern and the characteristic alterations seen in a disorder. And this procedure is referred by us as polyconnectomic scoring. So now I want to explain you how exactly we computed polyconnectomic score, or PCS. So to compute polyconnectomic score, we first need, as I mentioned before, a template of the alteration present in a brain disorder. This disease map or template we call in the paper connectome summary statistic, again taking inspiration from the genetic field. To compute the summary statistic, we first acquire MRI data from, let's say, patients and control, represented here in green and gray. And we measure the brain activity in gray matter regions that are analyzed during resting state, and estimate how synchronized is the activity between a pair of regions, allowing to reconstruct the functional connectome of a subject. This functional connectivity, oh, sorry, something get dark in the room. The functional connectivity is a matrix where rows and columns represent a brain region, and each element in this matrix is the functional connectivity strength between a pair of brain regions. Okay, so this is what we call functional connectivity or functional connectome. Next, for each connection in this connectome, we compare the functional connectivity strength between patients and control. This comparison will yield an effect size per connection that indicate the difference between the two groups. The effect size can be measured using COINS-D, Pearson's correlation, beta coefficient from linear regression, or any other statistical method that you want. The only important thing is that a higher effect size should correspond to higher functional connectivity in patients compared to controls. Okay, so the direction has to be like that. Positive effect size in the summary statistic mean higher values in patients compared to controls. Once we do this for all connection, we can generate basically the disease map of the disorder of interest or the connectome summary statistic. Once we have the connectome summary statistic, we can acquire data from a new subject. And then we multiply each functional connectivity value of this new subject with the corresponding connection in the summary statistic. And then we take the mean across all connections. So in other words, we compute the weighted average between the summary statistic and the functional connectivity of the subject, resulting in one single score that indicate how similar is an individual brain to the characteristic brain of the phenotype of interest that we are investigating. And this score is what we call polyconnectomic score. So it's extremely simple, this equation, let's say. Now, similar to the polygenic risk score, polyconnectomic score is a relative measure that must be interpreted by comparison to other polyconnectomic scores from other subjects. So PCS, polyconnectomic score, doesn't have a meaning, the numbers by itself. It has to be compared to others. So PCS should be computed in an entire sample of individuals. And then when you compare the individuals based on PCS, we can start gaining more meaning about what PCS means. So basically, higher PCS are those brains that resemble more the disorder of interest than those that have lower PCS. So in summary, this method involved three main steps. First, we estimate the disease map or summary statistic. Second, we calculate polyconnectomic score as the weighted average of the summary statistic and the subject's functional connectivity. And three, we compare PCS across subjects. Now, I want to briefly explain you a bit the data that we use for this study. For this study, we collected 22 independent clinical MRI datasets, comprising approximately 11,000 individuals across 11 neuropsychiatric and neurodegenerative disorders. Additionally, we conducted further analysis in 27,000 subjects from the general population, including both cases and control from the UKB Biobank. So in total, we have around 37,000 subjects that were analyzed in this study. Now, on the right side, I put the names of each dataset that were included in the study. All but one dataset are open access, so everyone can have access to this. And these datasets are primarily sourced from the USA, but also from Europe, Asia, and Latin America. So we try to be as diverse as possible. The data used in this study was resting state fMRI. We parcellated the brain to 68 brain regions using the Zika and Kiliani Atlas, which result in approximately 2,000 brain connections. We also regress out covariates from the functional connectivity data, including age, sex, site, and motion. So we make sure that none of these variables has an influence in our results. So now let's focus on how it looks the connectome summary statistic of a disorder. So here, in this case, I'm showing you the summary statistic of bipolar disorder. The three dots in the upper right corner indicate that we used 5 datasets, 1,790 controls, and 320 patients, from which we conducted a meta-analysis using all these available data to estimate the disease map or summary statistic that you're seeing in the screen. Now, I want to explain you a little bit what this means. So each element in the matrix represents the effect size of that connection, with orange, in this case, indicating negative effect size or hypoconnectivity in patients compared to controls. Gray, in this case, represent hyperconnectivity in patients. Above the matrix, there is a plot where the y-axis represents the minus log 10 p-value of each of these connections' effect size. So higher the value, the more significant is the effect. Now, I want to pay attention with the 3 different thresholds that we're showing. So if we apply Bonferroni correction, we found that a handful of significant connections associated with bipolar disorder reach this significant level. However, if we use an uncorrected threshold, in this case, the gray line, if we use an uncorrected threshold, we see there's a quite more broad range of subtle alteration that going on, which usually we will not even report in a neuroimaging study. So my question that I want to leave here, and I hope they're going to answer later, is: are these nominally significant connections, or even those connections that are non-significant, merely noise, or do they actually contain relevant information about the disorder? And here are the 11 summary statistics that we computed for our study. All these are resting state disease map, meta-analytic disease map, has been made available in the PCS toolbox, allowing anyone to compute PCS in their own data, which is something that I will discuss in the end of this talk. Now, just to clarify, in the subsequent analysis, we utilized the dataset with the largest number of patients as the summary statistics, and then we compute PCS in the other independent datasets to avoid sample overlap. Okay, so it's very important that always the summary statistics you're using, the sample of where you compute the disease map doesn't have the same individuals where you compute the PCS. Otherwise, you will be overfitting. It's not the idea. So yes, so as I said, the summary statistics shown in this picture are the meta-analytic one, and these are the ones that we made publicly available for everyone in the toolbox. So the first natural question is, well, if we indeed can distinguish patient from control using PCS, saying in another way, if patients indeed show increased PCS compared to control. So this is a bit of a complex figure, so I will break it down into parts, okay? So in this first example, we estimated the resting state summary statistic for ADHD using the dataset with the largest number of patients, and we included all function connections, so the whole brain. And then we used the summary statistic to compute the PCS for ADHD in three independent datasets, containing both patients with ADHD and controls. Now, each dot here represents one dataset, with the dot side corresponding to the sample size of that dataset. In the x-axis, we're showing the area under the curve of a logistic regression, predicting disease status of all the subjects in each one of these datasets using the PCS for ADHD. Filled circles indicate dataset where PCS was significantly higher in patients compared to controls as determined by a t-test. Okay, and those datasets that were significant, we show in the right side the histogram of the PCS distribution for patients and control from this significant dataset. So in this case that I'm showing right now, we found only one dataset showing significant differences between patients and control, where indeed ADHD patients show higher PCS for ADHD than control. What this suggests that the whole brain disease pattern that we observed in the summary statistic, derived from a completely different sample, were partially replicable in ADHD patients from this significant dataset. Now moving to other three disorders that typically exhibit early onset. So in this case, we have autism, OCD, anxiety beside the ADHD. We found that autism patients show higher PCS than control in two datasets, achieving an accuracy with an AUC greater than 0.65 in one of these datasets. So once again, we found that the whole brain disease pattern that are associated with autism observed in one sample, the summary statistic, were found to be present in autism patients in other two datasets. Now, I'm presenting you neuropsychiatric disorders that usually occur in adults. In this case, schizophrenia, schizoaffective disorder, bipolar disorder, and major depressive disorder. We found that all four disorders exhibit generalizable disease pattern in at least one dataset, with accuracy greater than 0.65 for schizophrenia and schizoaffective and 0.7 for bipolar disorder. Now, I would like to highlight once again the result of schizophrenia, where we see again that the whole brain disease pattern that we observed in the sample used to compute the summary statistic were consistently replicated in a schizophrenia patient across 3 independent data. Okay, so that gives us some certainty that indeed with this, there's some similarity in the disease pattern, at least in schizophrenia, and quite diverse samples. I want to also highlight that these samples include from patients from Japan, patients from Germany, patients from the US, different scanners, and sometimes even different age, a little bit different age. So it seems to be quite robust. Finally, we have our three neurodegenerative disorders that we investigated: frontotemporal dementia, Alzheimer's disease, and Parkinson's disease. These 3 disorders show at least one data set where we could find generalizable whole brain disease patterns. Notably, even in data set with a small sample size like this one, PCS can significantly distinguish patient from control, achieving high accuracy. So indeed, the sample size play a role, but still it had enough power to distinguish patients in control even with low sample size. And this is the complete figure. Okay, each of the histogram in the right side include the Cohen's d group difference in PCS between patients and control, and we found there are the range is quite variable from quite a small effect size of 0.17 Cohen's d for ADHD to larger effect size 0.9 for frontotemporal dementia. So quite a strong effect. Overall, we found that 9 out of the 11 neuropsychiatric and neurodegenerative conditions investigated exhibit this cross-cohort generalizable whole brain disease pattern. Okay, and in some cases, these disease patterns are replicated. Move on now. And now, as you can see, well, PCS is quite robust. It worked in 9 out of the 11 disorders, but didn't work with all the disorders. So what happened there? So we investigated different things. Now I only want to show you one factor. So one factor that impacts the accuracy of PCS, it seems to be the demographic differences between the sample where the disease map was computed versus the sample where the PCS is computed. And one of the demographic factors that affected the more was age. So here in the x-axis, I'm showing the mean age difference between the sample in the summary statistic with versus where PCS was computed. And in the y-axis, we have the area of the curve predicting disease status using PCS. So we can see that if the age distribution between these 2 samples differs a lot, PCS performance is likely to be reduced. So this correlation is of minus 0.4 Pearson's correlation. So to put it in other words, if the disease map of a children, let's say with depression, the disease map of a children with depression is likely to not be very similar to the disease pattern that we observe in adults with depression. Okay, if this is indeed the case, PCS will probably perform less better. Now, this is an important consideration when choosing your summary statistic. The sample hopefully should be as similar as possible to the replication dataset where PCS will be computed, at least in terms of demographics, unless that your goal is to test a specific hypothesis related to this, such as whether the disease pattern in younger population are replicable in older cohorts. Okay, so that's one note that I want to make sure to put here. Now, we wanted to compare the predictive power of PCS using and compare between a whole brain versus including only focal effects. So again, in polygenic risk score, researchers have shown that when we include variants that did not reach genome-wide significant threshold, still we can improve the prediction accuracy of polygenic risk score. So to give a quick example of polygenic risk score, and here is predicting bipolar and schizophrenia status using different summary statistics. And we can see that the prediction of PRS is quite improved when they include variants with relatively high p-values. So this suggests that even variants that are deemed non-significant in a GWAS may still carry relevant information about the phenotype being investigated. So we aim to apply a similar approach with PCS. So here in the main analysis that I showed you before, we conducted, computed the summary statistic, including the entire connector. So the all 2,000 plus connection. In this plot, each dot is a data set from each disorder. Y-axis is the area under the curve, again, predicting disease status using PCS. The filled circles are data sets where patients show significantly higher PCS than controls. And the one that only with a circle and a transparent inside are those ones that PCS was not able to distinguish patients and control. Now, we see that when we include all brain connections, the whole brain, we found that patients show higher PCS than controls in 9 disorders across 13 datasets. If we move forward and we want to threshold the summary statistic based on p-value, and we threshold using including only the nominally significant connection, so effects with a p-value below 0.05. When we do this, we found that around 60 to 300 connections are significant across the summary statistic of the disorders. Now, as you can see, using this nominally significant connection, we found that 9 datasets across 7 disorders show higher PCS in patients compared to control. So already we can see that when we include all connections, we had more data about the disorder of interest than when we include only the nominally significant connections. In other words, even non-significant connection seems to contain relevant information about the disorder investigated, and they are not simply noise. We can move forward thresholding of the summary statistic, and we apply network-based statistic. So NBS is a method that assumes that altered connections typically occur in group of cluster of interconnected connections. This topological assumption about the altered connections allows for controlling multiple testing in a manner that is less stringent than FDR correction, but more stringent than nominal correction. So if we apply NBS to the summary statistic, we found around 200 and 300 connections that survive this analysis in 6 disorders, not in all 11. That's why you see less dot in NBS, because 5 disorders didn't show any connection significant when you apply NBS. So when you threshold the summary statistic using NBS, we found 9 datasets showing group difference between patients and control across 5 disorders. Moving forward to a little bit more stringent p-value threshold, namely FDR correction of the summary statistic, this results in only 10 significant connections for Alzheimer's disease and 24 connections for autism. Again, we see less dots here because only 2 disorders had FDR significant connections. This highlights how already applying FDR correction will lose considerably information about the disorder. In this analysis, we found the 4 datasets show patients with higher PCS than control across both disorders. Now, I want to make a side note, and I want to highlight something that I find very interesting, that only with 24 connections, in the case of autism, we were able to distinguish autism patients from controls in 3 independent datasets. So this indicates that these only 24 connections appear to be consistently affected in Alzheimer's in autism patients. Okay, I found that quite remarkable. And finally, we have Bonferroni correction, which led to only 1 to 3 significant connections, again in Alzheimer's and autism. That's why we have the same number of dots than in the FDR. And we found only 1 dataset showing significant group difference in PCS. Okay, so overall, this analysis suggests that when we threshold the summary statistic based on significant levels, we lose considerable data. We lose considerable information, and even connection with non-significant p-value still contain valuable information regarding the investigated disorder. Information that is lost when we apply even a liberal nominal threshold. Okay, so yeah, any questions so far with this? For me, it's important that this is for everyone clear.
So for questions, you can just raise it with your microphone. If you don't have microphone at the moment, please type it in the chat box. I will read it for you. Yeah.
Okay.
Yeah.
Okay. There's no question. I cannot see the messages actually.
Not yet. So
okay.
If there are any questions, I will let you know.
Okay. So now let's move on to other applications that we can do with PCS. So far we've seen whether PCS can distinguish patients from control. Now PCS have several applications. One, in my opinion, useful application is to examine brain behavior association. So typically, these brain behavior associations are established by correlating each brain connection with a trait of interest. Now, the brain behavior association are involve relatively weak effects. And if we combine this with the high number of tests conducted, it makes it quite difficult to robustly detect such associations. So PCS has the advantage that aggregate evidence of disease signature within an individual brain into a single personalized score. So it's 1 score per person that summarizes all this evidence of alteration. So this approach enables a straightforward brain behavior correlation. So to show this, we compute PCS for each one of these disorders in the general population sample of the UKB biobank cohort, around 27,000. And we correlated each one of these PCS score with clinical, behavioral, and even body-related measurements. So in this plot, we group the disorders in 3. In the x-axis represent the Pearson's correlation between PCS and a variable. And again, the field circles indicate significant brain behavior correlations. Now, these correlations are indeed weak, which is not surprising, given that these are brain behavior association, but also we're using a huge sample. So that usually end up with lower correlations coefficient. However, we can observe common pattern emerging across disorder. You don't see my mouse, but in the upper corner, we have a cognitive measurement. So we see that those subjects with a higher PCS for neuropsychiatric disorders tend to show lower fluid intelligence and longer reaction times. Also below in the metric that we show below, we also find that that those with a brain more similar to neuropsychiatric disorders tend to show higher neuroticism-related measurements. Okay, so this is indeed within the line that we expected. Also with well-being factors, we see a lot of these PCS associated with lower health satisfaction, lower work satisfaction, lower happiness, okay? Now, also we see unique associations, okay? For instance, individuals with a brain more similar to those with depression or anxiety show higher BMI, which is quite interesting if you see previous literature. But also we saw that those subjects with a higher PCS for frontotemporal dementia, so those subjects that have a brain more similar to those with frontotemporal dementia show lower BMI. Okay, so this is one example how we can use PCS to uncover brain behavior correlation. Now, sky is a bit the limit. In this case, PCS has multiple applications, and I would like to just briefly mention a couple of them, okay, before moving forward to the toolbox. So one option is to do cross-disorder analysis. For example, here, we computed the PCS for autism in 2 multi-disorder datasets, okay? In the right side, we see a dataset with mainly kids and adolescents, and we found that only patients with autism show higher PCS for autism than controls, suggesting quite some degree of specificity for this marker. Now, in the left side is a dataset with adult patients, where both schizophrenia and autism patients show higher PCS for autism. This means that schizophrenia patients also show whole brain disease signatures related to autism, which is again in line with previous studies showing quite some overlap between the disease map and the genetics of these both conditions. Another example is patient stratification. Well, we have one of our data set, very nice, called BSNP, have patients across the psychosis continuum, but also have relative of patients with schizophrenia. So here we found that schizophrenia patients display the higher PCS of schizophrenia across all these samples, but it was significantly higher than the PCS of their first degree relatives, which we know they already have a high risk to develop psychosis. At the same time, we found that those first degree relative schizophrenia patients also show higher PCS of schizophrenia compared to control. So we can see, as expected, nicely, an increase in the disease signature related to schizophrenia, going from controls, healthy controls, to relative or schizophrenia patient, and with the highest with the schizophrenia patients. Okay, so today I explained the methodology behind PCS, and I discussed several applications, including distinguishing patients from control, detecting brain behavior correlation, and we briefly touch upon covering cross-disorder analysis and patient stratification. Now, to enable researchers to compute PCS on their own data and apply it to the different applications that we presented, and also potentially to expand it to other applications, all the scripts to compute PCS and all the disease map have been made publicly available on GitHub. So we made publicly available meta-analytic disease map of 11 disorders across 22 independent datasets. We use around 5,000 patients and 5,000 controls. And here I'm showing the demographic table of these disorders. And also you can see the number of datasets used to compute the meta-analytical summary statistic that I showed you in the beginning of the presentation. Now, one of the things that something that we encounter is, like, if you want to compute PCS, well, everyone has their own favorite atlas, and the resolution basically how you parcellate the brain, is crucial. So the summary statistic have to be parcellated with the same atlas where you want to compute PCS. So we map the summary statistic across 25 different brain resolutions. As I mentioned, again, this is essential because if you want to compute PCS in your dataset, our summary statistic must to be in the same atlas that your data. So our goal was to include a wide range of atlases and resolution so everyone can compute PCS on their own data. These atlases included a quite broad selection, including cytoarchitectonic atlases, also general-based, functional, and multimodal atlases. So you have basically a big menu that you can choose to compute PCS on your data. Finally, the scripts were made publicly available in R language, in MATLAB, and in Python. Okay, so everyone can use it in their own favorite programming language. Now, I would like to show you a bit how the main function of this toolbox work. So the main function, calculate PCS, take as an input a two-dimensional connectivity matrix of an individual, or also can be a three-dimensional matrix with connectivity data from multiple individuals. Okay, so you can have one matrix or group of matrices in one variable. And then the disorder parameter allows you to select which disorder to use as the summary statistic from the 11 available disorders. Disease map were computed using both with and without global mean correction, which can be selected using the G-mean parameter. And finally, we have the atlas parameter, which specifies the desired atlas that you want to use from the list that I showed you before. Okay, so it's relatively easy to compute PCS using this toolbox. Now, there's two key points that I want to highlight to make this tool as effective as possible. So first, we recommend that if your functional connectivity data was computed using global mean correction, we advise that the summary statistic should be also with global mean correction, okay? But more importantly, the summary statistic must to align with the same atlas as the one used by the user functional connectivity data, and with particular attention to the order of the regions, okay? So if the order of the region in your connectivity matrix is different than the order of the region that we have in the summary statistic, then the resultant PCS values will be meaningless, doesn't mean nothing. Now, to facilitate this alignment, each one of the disease maps in the GitHub, in the toolbox, include the region name in the matrix index and columns. So that allows you to verify what is the region order of the summary statistic, and then you can align this order to your own data, okay? So I think these are only the two main things that you should pay attention to work with PCS toolbox. With this, well, more information you can find in the GitHub page. You have the link below. Also, our paper was recently published in Biological Psychiatry. There we show all the details, the supplementary materials. Also, we have a lot of information there if you have any doubt. But also, if anyone has any question on how to implement PCS, please feel free to write me an email, which is down below, and I will do my utter best to guide you through your question. Okay? And with this, I would like to finalize this presentation. Thanks all the co-authors of this work, my colleagues at the Complex Trait Genetics Department, and more especially, I want to thank you for listening to this presentation.
Yeah, thank you, Ilan. Thank you for the excellent presentation. I really enjoyed it. The content was comprehensive, and the visuals, I have to say, were very engaging. So I will give others a moment to reflect. So I will